Membrane transport and facilitated diffusion of the phospholipid bilayer cell membrane. The cell membrane is what we call selectively permeable, which means it's really selective on what it'll let through, but it's permeable to some things. Some substances that can cross it more easily than others are really, really tiny. These are small, non-polar, hydrophobic molecules, things like O2 gas, nitrogen, N2 gas, carbon dioxide, and lots of hydrocarbons too. If they're small enough, they can just cross the membrane willy-nilly. Things that can be difficult to cross the membrane are going to need a little bit more help. They're going to need these proteins to assist them coming across the membrane. So these hydrophilic, non-polar, very, very large molecules and even ions, because they're small, but they have a charge, so they interact with things, they're going to need some more help. These are things like water, because it's polar, sugar, because it can be very, very large. So when we're trying to move these items across the membrane, we can do it in two different ways. The cell membrane has a passive and an active transport system. Passive transport essentially means it does not require energy. So a passive transport is when a molecule can use, I don't know, like a protein of sorts that doesn't use a lot of energy. A lot of times it will also move a concentration gradient. So there'll be some sort of like high concentration, low concentration factor that will allow the molecule to move in or out of the cell. So this is very important because this is really how a lot of materials and how cells export their waste. So examples of passive transport you might be familiar with are diffusion, osmosis, and then facilitated diffusion. When something moves down a concentration gradient, just spontaneously resulting in molecules changing their concentration, that's just called diffusion. It's a generalized term that we use. Substances are always going to move from like a high concentration to a low concentration. That's why we call it moving down the concentration gradient. Think about it in terms of this. If someone's over in the corner of the room and they spray perfume, perfume is a high concentration over by that person. So the perfume molecules are they're aromatic, they kind of bounce around and move, and they're going to move from the person who sprayed it, and eventually across the other side of the room, someone is going to smell it. They're moving from high concentration to where there was little or no concentration. Remember that the membrane is still, like I said, selectively permeable. So molecules are going to move across it, diffuse, if you will, at different rates. The fluid on the outside or the cytoplasm on the inside definitely affect this process depending upon where the concentration is allowed to be higher or lower. I always find that students find osmosis the most difficult, but it's real easy when you just remember osmosis. We're talking about the water. Where's the water molecule moving? Follow the water molecule. That's how we change like size and mass really quickly. Osmosis, the diffusion of water, it is going to move down its concentration gradient against that selectively permeable membrane. It can also be thought of as the diffusion of water of areas from that low solute concentration to areas of that high solute concentration. So essentially it's going to work a little in reverse from diffusion. Water is going to move to dilute what's concentrated. So in my example here, I have it's more concentrated on the right-hand side than it is on the left-hand side. So the water molecule itself wants to move to dilute what's on that right-hand side. So we see the volume of water on the left-hand side decreasing and the volume of water on the right-hand side increasing as water moves against a selectively permeable membrane, selectively permeable to water. It moves to dilute. Facilitated diffusion is diffusion of molecules through those membrane proteins that I talked about earlier. They're transport proteins. You can increase the rate of diffusion for things that are like small ions, water, and carbohydrates. There's really two categories of these transport proteins. There's what we call channel proteins and carrier proteins. Each transport protein in a very specific way, and it's specific for certain substances, but it facilitates the movement for passive transport. So why is facilitated diffusion passive transport and it's not an active transport? Why is using that protein not considered active or using energy? And that is because it is still a passive transport system, because the proteins are just assisting these things move down their concentration gradient. They're not using any energy to do this. I like to think of facilitated diffusion. Do you remember those toys that you had when you were younger? Maybe you have a younger brother or sister or niece or nephew. They have a ball, right? It's a ball. And on the outside of the ball, there's all these different shapes. Like there's like a star shape, a little circle shape, a little square shape. And you give it to a baby. And the baby has to figure out how to, put the star shape through the star shaped hole. And it's not requiring any energy in terms of, there's no mechanism. It's just a plastic ball and it's just plastic toys, but it provides such a specific doorway that it allows an extra assisted way for things to move down a concentration gradient. It's almost like a perfect doorway, a perfect opening for whatever that protein's looking for. So these channel proteins, they provide a channel for molecules and ions to pass through. And this channel is hydrophilic. Many are gated and they have a certain system to them. They only allow passage when there's some sort of like stimulus or they're prompted somehow. There's such a thing as aquaporins. So they allow they're a specific protein channel and they're just special for water. And if you think about it, you're like, well, why do if water moves, concentration gradient as osmosis moves, what does it need a protein for? But these aquaporins are they increase the rate at which this happens. So it's like a high speed water slide for water molecules, easy access in or out of the cell. Carrier proteins, they undergo these conformational changes for substances to pass. They almost have a working door locking system. So the one side, the binding site, is open, and then this protein goes through this conformational change where it closes and opens on the other side. So the protein is assisting in the movement of substances to pass through. These conformational changes, the best way I have to explain it is that it alters its secondary, tertiary, and even quaternary structure internally without altering the overall shape of the protein. All right, so let's practice. In terms of diffusion, describe what is most likely to happen to the particles in the image below. In regular old good old-fashioned diffusion, things move from high concentration to low concentration. So when these particles are diffusing, this particle right here is going to move across the membrane until we reach a status of equilibrium. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16. So there's 16 total. So there. It's going to move so that it finds a equilibrium. So it's going to go from that to that. In biology, we always try to find a way to illustrate a concept, model a concept, because a lot of this stuff you can't really, it's not like you can peer internally into a cell and watch the ions change place. So we're using models and descriptive laboratory type of things in order to understand what's going on. So it helps to draw to better understand what's going on such a small level. So why do you think energy is not required for facilitated diffusion? Why would a cell have options where it's not requiring energy in order to move things across its membrane?